come in so that we can get started. All right, welcome everyone to the second contributed talk session. Our first speaker is Tiasha Shaha Roy from the University of Minnesota.
Good afternoon, everyone. Today I'll be talking about our ongoing work on the relationship between vision and mental imagery. Most of us experience mental imagery as an approximation to vision. Reacquaint yourself with this familiar painting. In case you haven't seen it before, you can just take a good look now. Now, I'm going to remove this painting from the screen and ask you to imagine it. If you had to describe your subjective experience of the imagery, only a few of you would report that your mental image was as vivid or as detailed as the image you had seen on the screen. Some of you might even report that you had no mental imagery at all, while for the most of you who are like me, you would say that your mental image kind of resembled the scene image, but was not exactly like it. So it didn't feel exactly as real, for example. So what does this approximate nature mean for how mental images are encoded in brain activity? For many years, a consensus model has been that mental imagery is like weak vision. Well, that is a very intuitive description of mental imagery, since it seems to capture the subjective experience of it. We do feel that mental imagery is like some kind of dimming of vision. It is also consistent with the fact that brain activity during acts of mental imagery are characterized by low signal-to-noise ratio. So what do I mean by signal-to-noise? It's the ratio of the signal variance to the noise variance. The signal variance is the variance across trial average brain activity for different scene and mental images, while noise variance is the average spread of brain activity across multiple repetitions of the same scene or mental image. So as you see in this figure, the SNR for vision, which is denoted in blue, is remarkably higher than the SNR during imagery, especially in the early visual areas. So weak vision does seem to be a probable explanation for these observed phenomena. However, weakness as a description of mental imagery is rather underspecified and leaves out a lot of what we have learned about imagery in the last few years. For example, one thing that our lab and others have shown is that mental images are represented with lower spatial resolution than scene images. In 2020, we performed an experiment where we scanned human subjects while they saw and imagined in separate runs the same 1,000 or more natural image patches. We were able to estimate the receptive fields and the spatial frequency preferences for individual voxels during both vision and imagery. We found that the imagery receptive fields were larger than the visual receptive fields in the early visual areas. We also saw that the imagined spatial frequency preferences were lower than the scene spatial frequency preferences. These findings indicate that mental images are not just weak, but also lossy. And this inspired us to think of an alternate explanation or a description of mental imagery as a compressed form of vision. So what do we mean by compressed vision, and how does it differ from our usage of the term weak vision? Consider that brain activity lives in a 2D voxel space where distance along the x-axis here is your activity in voxel 1, and distance along the y-axis is activity in voxel 2. So if I have these cyan dots represent the brain activity during multiple repetitions of the same scene image, then we can have a blue dot at the center denoting the trial average brain activity, which we take it as a proxy of the visual signal. According to the weak vision model, The imagery signal is a scaled-down version of the visual signal, and in this case, the orange dots denote the imagery activity for different repetitions of the same image. So we have two other scene images and their corresponding mental images. For the compressed vision model, we say that the imagined signal is basically the visual signal in a lower dimensional space. In both of these cases, we have simulated the data such that the relative signal variance of imagery is constant. However, the key difference here is that in the first case, that is the weak vision case, the imagery signal varies along 2 dimensions, same as the visual signal, while in the second case, the imagery signal varies only along 1 dimension, while the visual signal varies along 2 dimensions. So to test or to find evidence in favor of either of these hypotheses, we need to be able to model the transformations between vision and imagery. And if the compressed vision model were to be true, we think that a transformation that compresses the visual signal should explain imagery signal as accurately as one that doesn't. And there should be fewer dimensions of imagery signal variance than there are dimensions of visual signal variance, at least in the early visual areas. So we did an experiment to test this, and this data set was collected as part of the natural scenes data set effort. 8 human subjects learned to associate each image with a single letter Q. For example, H for a horizontal bar, V for a vertical bar, and so on. There were 6 simple images consisting of bars and crosses and 6 natural scenes. So each of these images were repeated 8 times during a run, and there were 2 different kinds of runs, vision and imagery. During the vision runs, subjects saw images on the screen and a target letter Q, and their task was to say whether the target image matched the image that they were seeing on the screen. For the imagery task, they had to imagine the target image and project their imagined image on the gray screen in front of them and say whether their mental image was vivid or not. So once we have the visual activity and the imagery activity, we want to have models that convert one to the other. So we want to have models that transform the visual activity to the imagery activity. So we use what is a voxel-to-voxel modeling approach. We had used this approach previously in another work where Maggie was the leading author. So in the simplest case of this approach, we have imagery activity in one voxel being predicted by visual activity in the same voxel by a simple scaling like operation. So think of it as a simple linear regression. So, can you see my cursor? So this is, for example, the voxel here, and so on. So I predict each imagery activity during at each voxel using the visual activity at the same voxel. So this model basically mimics the weak vision case. And now, to test the compressed vision, we introduced a slightly complicated model. In this approach, we tried to predict activity in every voxel during imagery as a linear transformation of the activities of all voxels during vision. Importantly, we impose a linear hidden layer of dimension that is less than or equal to the number of input or the dimension of the input or output layers. In this case, the number in the input and output layers is the number of voxels. So by constructing such a mapping, we obtain a linear autoencoder. And we fit both of these models and assess the cross-validation prediction accuracy of each. On this figure on the x-axis, we have the model advantage of the compressed vision model, hypothesis relative to the simple vision, relative to the weak vision case. While in the y-axis, we have the average model performance. If compressed vision was in fact a reliable model, we would expect a slight improvement in the prediction accuracy. And here, each dot corresponds to one subject and one ROI. So we see that the compressed vision model does performs like better in most cases. And this kind of puts a check mark on our first prediction. Next, we want to be able to estimate the potential reduction in dimensionality from vision to imagery. In order to do that, first we have to know the dimensionality of the visual signal variance. So we place a compressed vis-to-vis model to estimate the visual signal dimension. And we find the dimensionality using the validation set. Here in this case, for example, this is an example case where we have the dimensionality of the visual signal to be 10 for V1 of a subject. Next, we want to see the dimensions of visual activity along which there is predictable imagery variance. And we also use a similar process. We use the validation set to assess it. And here in this case, it is 3 in case of imagery, denoted in red. Now, if we plot the overall dimensionality that we estimated from our compressed vis-to-vis and vis-to-imagery models, we see that in the early visual areas, the number of dimensions of imagery signal variance that can be accurately predicted from visual activity is fewer than the number of predictable dimensions of visual signal variance. To tie this back to our original inspiration, remember that it was in the early visual areas where we saw a dramatic reduction in the SNR and also where the spatial resolution was lower. So it makes sense that it is in the early areas where we would see this relative reduction in imagery dimensions. So this is the SNR plot that I had before. It looks quite similar. And this also puts a check mark on our second prediction that there should be fewer dimensions of imagery signal variance than there are dimensions of visual signal variance, at least in the early visual areas. To conclude, we find that empirical evidence, along with our current data set, lends support to the compressed vision formulation of mental imagery, and a model that explicitly reduces dimensionality of the visual signal fares better than one that doesn't. We have fewer visual dimensions that explain variance in the early visual areas during imagery. And we wonder what specific dimensions of visual coding might be preserved during imagery, and why might the brain choose to preserve some coding dimensions while tolerating the loss of others. With that, I'd like to thank my mentors, Thomas and Kendrick, for their constant support and guidance, Jesse for collecting this data set, Ghislaine for his theoretical insight, and all my lab members from the NASA Laris Lab and CVN Lab for their constant encouragement and feedback. And I thank you all for paying attention to my talk.
Thanks, Tiasha. We have time for a question.
So thank you so much. I thought this is really amazing work. It's really, really cool and insightful. I was wondering if you think that we could create a visual stimulus that is so much compressed that it becomes indistinguishable from imagination, or do you think that this compression is just inherent to the neural signal, that no matter how degraded your visual stimulus is, imagining it will always degrade it further just because of the brain? Does that make sense?
So you want to make a change in the pixel space. Yeah, that is a good idea. We could explore if we could lower the contrast or something of the image and try to, or maybe compress the image, but whether that exactly relates to a compression in the neural signal space, which is nonlinear, might not be the case. So, but however, I do not know what it would be. We would have to do some experiments to explore that.
Thank you.
That was a good question.
We can have one more quick question while the next speaker sets up.
Okay, sorry.
Thank you for the great talk. It was really inspiring to me that you predict mental image from the actual visual voxels. Like, I wonder if it's possible to do the opposite, like you take the mental image and try to predict the visual voxels.
We haven't tried that because mental image signal is inherently a little more, has low SNR. We haven't done that. We could try to do that, but in this case, as you saw, we had only 12 images. If we probably had a lot more images and a lot more repetitions, that would be probably a successful attempt. But we do not like at this point, what we want to explore is the number of dimensions of visual signal variance that can predict imagery. Because vision is more like, that's what we are seeing, that's the input signal, that's the main thing. But are you asking about the dimensions or just the prediction?
I'm just trying to say that you use a low dimension, try to predict the high dimension stuff and see what can be predicted. So the part can be predicted, can be that part kept in this low dimension.
Sorry, what? I could not hear. What can be predicted?
So sorry, I just mean like you have this low dimension mental image space and high dimensional actual visual image space. If some part can be predicted from the low dimension space to the high dimension space, that means this part is kept in this low dimension space.
Yeah, we could try that. Interesting. Yeah.
Yes, thank you.
All right. Next up is Kushan Mukherjee from the University of Wisconsin-Madison.
Right. Cool. Hi, everyone. Thanks for coming to this talk. All right, let's get started. So at CCN this year, right, we've been grappling with these questions about the brain, such as what does it mean to really understand the brain? Are artificial neural networks kind of good hypotheses for brain function? And like, what's the kind of question that deep learning is often answering? And maybe like what's really important? Architecture, training diets, objective functions, learning algorithms, and this applies to whatever your favorite type vision model is, be it classic CNNs, more recent vision transformers, or even more recent kind of CLIP-style image-language contrastive learning models. What I'm going to say is, for this talk, we're just going to put a little box around these and just maybe put a pin in it for a second. And for two good reasons. First is, at least in some way, almost all of these models have been falsified, if not all of them, on some empirical benchmarks. And secondly, we know that the primate... Oops. Okay, it's showing up. Okay. Yeah. And secondly, we know that the primate brain is complicated and that we're at least a bit far away from having like a perfect model of the cortical architecture. And so here I'm actually going to argue for and hopefully motivate that these models are still useful because of this kind of classic quote from George Box that we all probably know, that all models are wrong, but some are useful. And I'm going to argue that maybe we should be thinking of usefulness in ways that go beyond just predicting behavior or thinking about neural fits. Okay, so I'm being coy about it, right? Like, useful for what? So maybe motivate it. I'm going to talk about this particular task, which is graded emotion recognition. Right? So this is kind of like a paradigm we adapted from Wang and Adolph's, where you are shown a face, you're immediately asked to make a judgment about whether, say, it's fearful or happy, and then you're asked to maybe give a confidence rating of how confident you are of the rating you gave. We're going to focus on maybe just this first part of the emotion discrimination judgment. So if you ask a bunch of people to do this task, you see data that look like this, where on the x-axis you see faces ranging from maximally fearful to maximally happy, and on the y-axis you see the kind of probability of someone saying that it's a happy face. So this is a nice little psychometric curve which shows that there's kind of good sensitivity, but this isn't really the case for everyone. So if you look at a particular subpopulation, so that's specifically that of adults with autism spectrum disorder, or ASD, you can see that the kind of psychometric curves you see here are a bit shallower, right? And in other words, there's kind of reduced sensitivity in emotion judgments in this kind of paradigm. So what this means, again, more concretely, is to elicit a kind of behavioral difference of at least this much. For neurotypical populations, you'd only need to kind of morph a face this much on the x-axis. But to get the equivalent amount of behavioral difference in, say, autistic populations, you'd need to at least morph the face that much. And I want to really emphasize that these kinds of differences in behavior can't be attributed to something like, variations in gaze patterns when participants are looking at these stimuli. Because, in this data that we again adopted from Wang and Adolph's, they made sure to really control using eye tracking where people were looking at on the stimuli when making these judgments. So those are kind of really controlled for between these two populations. So that kind of leaves this open question on the perceptual side, what's really going on that might be beginning taking into account for this difference? One kind of salient axis is that instead of looking at this data in this kind of aggregated fashion, where we kind of collapse data across all participants and all images, one thing that might be interesting to think about is image by image variation, which might give us a kind of nuanced picture as to why this kind of red curve is a bit shallower. So specifically here, I'm plotting the same kind of emotion happiness scores made by control neurotypical people on the x-axis and by autistic adults on the y-axis. And here you can see that for many of these images, they're actually quite close to this identity line. But there are a couple that are kind of like removed. And these are the images that elicit the biggest differences between these two groups. And it's in fact images like the one that I just highlighted in red that we really care about, because they're the most helpful in diagnosis in kind of finding differences between these two groups. And so what we want to do is find a principled way of finding out a priori what these images might be. In other words, we're asking how can we generate useful image level hypotheses about what can kind of find the biggest differences between these two groups. So the first ingredient to any kind of image level hypothesis is having an image computable model. So in this case, we use artificial neural networks, many of which nowadays have kind of mappings between the layers of the model to layers of visual cortex, such as this. But since we really are talking about usefulness, and specifically the usefulness of the latent representations learned by the model, we can kind of use pre-trained CNNs, vision transformers, or CLIP without thinking too much about kind of biological plausibility. Right? Okay, so there are kind of three main kind of desiderata that we need to kind of tick off when we kind of embark on this research program. The first of which is, can artificial neural networks kind of perform this task the way that people do? The second, again, thinking about usefulness, is can artificial neural networks predict which images will be the most diagnostic in finding differences between these groups? And then finally, maybe looking ahead a little bit, can artificial neural networks help mitigate the phenotype or the differences that we observe between these groups, which might be kind of translated for treatments? Okay, so let's start with this first kind of problem of can these networks even do this task? Well, one kind of simple way to test this is to take pre-trained networks and fine-tune them by learning projection weights from the latent representations learned by these models to predict the kind of scores, these kind of happiness scores that we were just talking about, made by actual human participants in this kind of paradigm. And so what we do is we take a bunch of models and then we try to predict the scores. And I'm just going to show you one representative model. And here you can see that we can kind of recreate the psychometric curve that we saw from people in a few slides earlier. So this is like a good sanity check. And it tells us at the very least that these models, and we did this in the kind of right cross-validated way, so it's not kind of like overfitting or anything like that. And so it tells us that ANN models can indeed kind of predict facial emotions and can do this kind of discriminative task in the same way that people can, qualitatively at least. Okay, so a check on that front, great. So moving forward, can we use these models to predict which images are going to be the most diagnostic? And so we're going to take this paradigm we started to build up of using artificial neural networks. And so we're going to take a particular data set, so this data set of facial images, again, varying in emotion from fearful to happy, used by Wang and Adolphs, and we're going to fine-tune our model to predict two different things. So we're going to train a pair of models. The first model is going to predict the kind of emotional discriminative scores made by control neurotypical populations. We're going to train a separate model using the same neural network backbone, but this time to predict the ASD scores, right? And now what we're going to do critically is freeze these weights and now swap this data set out with a new data set of completely new images. And we're going to pass these through our model and generate predictions about what the control score should be, what the ASD score should be, with the critical goal of finding the difference between these predictions. And this is actually going to help us select images. And how might we do that, you ask? Well, really simply, we're going to just select images from this entire data set that have the highest delta or are predicted to have the greatest differences between these two groups. Right? And so there are three ways this can turn out. One, that the kind of images that we select in this model-based way end up being kind of just as diagnostic as random images you subsample from this data set. Second hypothesis being that we're actually worse doing this kind of model-driven way than just randomly selecting things. And finally, the last hypothesis is that we're actually going to do better using kind of ANNs to pre-select images. So what do we find? Well, we collected data on 80 new images from 12 new ASD and control populations, subjects. I really want to shout out Nayeon Kim and Ralph Adolphs, whose lab really kind of did the heavy lifting of collecting this data in the lab. And then what we find is a kind of interesting set of distributions, right? So the green here shows images that we selected using our model-driven approach, and gray bars are random images. And what you can see is that the distributions are shifted such that the green bars are shifted to the right, and in other words, that tells us that we actually get greater observed differences when we use model-based kind of feature selection, or image selection. And here are kind of averages of those two bars, and the differences between random and model-based selection is significant, which tells us that, you know, ANNs can indeed be used to predict which images are the most diagnostic. So we do find support for this kind of last hypothesis. Great. The other thing I want to really highlight, which is really interesting to us, is that the degree to which a model is good at selecting images that are going to be diagnostic of finding differences between these groups is strongly correlated with the ability of these models to even predict the neurotypical scores of people to begin with. So this is actually great because it shows that ANNs with better alignment to human behavior, or neurotypical behavior specifically, produce more accurate diagnostics. And this should hopefully kind of really incentivize us to really focus on building better models, not just for better brain fits or better, you know, specific hypothesis testing, but also to think really about the kind of translational effects of this. I'll talk more about this in a second. Okay, so ANNs do predict which images can be most diagnostic. Great. So moving on to this final kind of like, and you know, I know people like to work on their laptops during these talks, and if there's one thing I want you to take away from this talk, it's going to be the things that follow. So yeah. So one way to think about this, specifically about treatment, is, you know, given an image, we find differences between these two populations. What could we do to these images to mitigate those differences? In other words, in the counterfactual world, how would this image have to look so that a neurotypical person and an ASD person would kind of converge on their scores a bit? Right. So for example, here, this is one particular image. We can pass it through our pipeline, get predictions of the differences, and we'd want to really minimize this delta between, you know, control and ASD scores. So here's what the kind of actual differences between these two populations when asked to do the kind of emotion discrimination task on one set of participants. And so we really want to bring that number down to 0 as close as possible. How can we do this? Well, luckily, we live in an age when, you know, we have really powerful generative models. So what we did was we leveraged a generative adversarial network that was specifically trained to generate faces of varying emotions, and we used gradient descent to iteratively update the image in pixel space so as to get like a difference of 0 between the control and ASD scores. And so if you pay attention to this image, this is how it started off, and at the end, after our GAN did its magic, this is what it looked like. Okay, so we take this face, and now we show it to a new cohort of participants, and we ask them to, you know, of ASD and neurotypical participants, and we ask them to do the emotion discrimination task. And what do we find? Well, we find that the score does indeed go down, which tells us that, yeah, the perception of the emotion is changing across people. Here's another image. You can see that we did the whole generative image synthesis process again, and again we see a reduction in the overall difference between these two groups. We did this for a bunch of images, and on average we did find that there was like a reduction in the difference, but you know, you can see that there are a couple of images for which we didn't get kind of significant reduction, and you know, this is maybe not that surprising because I'm bringing back this whole plot where if you look at the x-axis, you can see that even our best model, CLIP, wasn't perfectly diagnostic of predicting even neurotypical behavior. So again, this is to go and say that once we have kind of better models or more rich latent spaces that can predict neurotypical behavior better, we might also see gains in this kind of image synthesis front. So for people who are building the next gen of models, build better models. Okay, and yeah, and this is also, you know, and so I looked at individual images, but on average, you can see that these differences are kind of brought close to 0, and it's a really significant effect. Great. So this really tells us that ANNs can also help mitigate the kind of differences between these groups, and that's really promising for treatment. I just want to contextualize this entire project in terms of kind of research program led by Ko, who was the kind of the guiding light behind this project in his lab, where there's kind of 3 main axes, right? The first is running kind of behavioral studies with primates and humans in the lab, and and online. Secondly is kind of getting neural measurements using invasive techniques. All of this in kind of support of building hypotheses and testing hypotheses in terms of computational models. And hopefully I've made a case today that kind of doing this kind of basic science has this kind of nice synergy with kind of translational issues, such as, let's say, in clinical settings where you can kind of build better diagnostic tools, better treatment tools, and as we kind of run more behavioral experiments, get better neural data to refine our computational models, those in turn can feed back and help build better diagnostic and treatment tools as well. And if that's not useful, I don't know what is. So with that, I just want to thank everyone, and I'm happy to take questions.
All right. We have time for one, maybe two questions.
Okay, thank you very much. That's a cool research direction. I have a question about your last part where you synthesize the image. So it seemed like the emotion was caricatural and super, super obvious. So isn't that a confound? So shouldn't you maybe try to regularize it so that it's not allowed to make the emotion too obvious?
Yeah, I mean, that's a great question. I mean, again, what I wanted, like, maybe that doesn't come across in this talk is that there were kind of like really image by image kind of variations as to like the features that were accentuated. So like, you know, in one example I showed, I think like the eyes got like more like, you know, widened, and like, you know, and other ones you'll see that the eyes don't change at all, and it's more of like the, you know, like the facial expression, right? And the fact that they seem caricatured, I guess, is kind of the point, right? It tells us that in terms of behavior, that's exactly the kind of direction that you need to move the images in like a principled way, using like a generative model to kind of mitigate the kind of differences. You know, otherwise we'd be thinking like doing ad hoc things like, oh, maybe I think I can, you know, make it more pronounced, but it's not immediately obvious how to make things more pronounced. But using a generative model gives us a kind of principled way to do that.
One more quick question.
Yeah, great talk. Yeah, is the delta that you're measuring between groups, for images that have a large delta between groups, do they typically have a large delta within group, like a lot of disagreement within group?
Yeah, so I think like the amounts of the delta between groups is definitely much larger than the kind of delta within groups. And so, you know, we've actually kind of replicated this not just within neurotypical populations in the lab, but we've also like, you know, run studies on prolific, and you can see that the data collected on prolific and in lab also kind of really strongly correlate. So the kind of variation in delta is actually really small within the groups, and it's only kind of when you look across groups that you kind of see those differences that we try to mitigate.
Thanks.
All right, let's thank Cushion again. All right, our next speaker is Jacob Prince from Harvard University.
Thanks, Layla. Not sure where the slides are. Yeah. All right. Not really prepared to improvise the whole talk. There we go. All right. All right, so classic framing of the problem of vision is that patterned light hits the retina and is transduced into neural signals and transformed through a series of cortical stages into a representation of the world. And the question we want to understand is, how does the visual system turn this input into a format that's useful? To make progress here, there's been a heavy focus on high-level visual cortex, which we know represents information about all different kinds of objects. And lately, deep neural networks have proven quite useful for studying high-level vision, since we can train models that vary in their visual diet or architectures or learning goals, and study how these pressures impact the representations they learn. And one of the major points of tension in studying these representations is whether the visual system tends to rely on representations that are more sparse or more distributed. And one way to approach this question is by recording responses of many neurons to a whole bunch of images to try to assess their rich response structure. We can think of this as a high-dimensional space where each neuron is a dimension and each image activates some neurons to some degree. And now we can try to understand the structure in this space so we can see if points are more clustered together or more separate and chart where the useful information lies. And one popular approach for this, of course, is principal components analysis, which basically highlights the major dimensions of variation in this feature space. So on the sparse coding extreme, we can imagine a toy population where every neuron is tuned to a completely different stimulus attribute. And here, every neuron is doing something different, something completely independent from every other neuron, and this would be a maximally sparse or efficient code, where the eigenspectrum is flat and high dimensional. And this means that each dimension of the principal components is accounting for roughly the same amount of variance. So these sparse codes are super efficient because there's no redundancy, but as a result, the overall population response could change pretty dramatically from small perturbations of the input. And on the other side, having a more distributed code would mean there's a lot more systematic variation and correlation in the responses, with neurons responding to many different images to different degrees. And in this scenario, responses along a few major dimensions might capture most of the useful variance, with the other dimensions of the code accounting for much less. And this redundancy ensures robustness and makes readout easier, but at some cost to information capacity. And people mean a lot of different things when they say distributed coding, but here we're just defining it as these kinds of correlated responses in the population with relevant information spread all across it. So this sets the stage for how these different extremes of neural population coding could capture the rich space of natural images, and these schematics were adapted from the work of Stringer and colleagues, who proposed that population responses are optimized to strike an ideal balance between the benefits of efficient high-dimensional codes and robust low-dimensional codes. And in this work, we asked whether we could use deep neural networks to operationalize and study this tension in a controlled way. In particular, our goal is to systematically manipulate the dimensionality of deep net responses. And to do so, we'll perform another variation on the theme of controlled model rearing, where many studies have examined the role of visual diet architecture and goal, but here we'll be holding all of these constant and putting a relatively underexplored inductive bias to use, which is regularization. In particular, we'll be studying a group of models that are trained with different amounts of dropout. Dropout was introduced around a decade ago as a technique to help prevent models from overfitting, where during training, random groups of units are temporarily deactivated with probability p, as you can see here. And this should prevent overfitting and improve robustness by ensuring that no single neuron becomes overly adapted or reliant on its specific inputs. So what's the consequence of dropout? Intuitively, we can think of it as pressuring the model toward learning smaller weights, since the random removal of resources means it would be risky to depend too strongly on any one connection. And as such, increasing dropout probability should pressure the network to learn more distributed representations of category information during ImageNet training, with more correlations in feature tuning and lower dimensional representations with steeper eigenspectra. So the rest of the talk will be split into two parts. First, looking at how dropout impacts ImageNet-trained deep nets, and second, asking what these results can tell us about human visual cortex. We first wanted to validate our intuition that increasing dropout would reduce the dimensionality of learned representations. So here we're going to use AlexNet, trained on ImageNet classification, with dropout applied at fully connected layers 6 and 7 toward the end of the network. We trained 10 models with dropout probabilities ranging from 0 to 0.9 in increments of 0.1. And of course, there are many different ways to quantify dimensionality, but here we followed the approach of Stringer and measured the slope of a linear fit to the log eigenspectrum. So how does dropout impact dimensionality? Here I'm plotting PC dimension on the x-axis and variance explained on the y-axis, and examining layer FC6 representations, and we can see that increasing dropout pretty systematically reduces dimensionality, with no dropout providing a shallower alpha of 1.01 and greater dropout raising that value to 1.7. And here's a summary of all the models' rates of eigenspectrum decay showing the trend. So dropout does seem like a good knob to manipulate dimensionality. And next we look deeper to see what other interesting things we might be able to learn from these models. I'll briefly highlight two different analyses. First, looking at task performance on ImageNet, showing top 5 accuracy as a function of dropout. This is the model with no dropout, and somewhat surprisingly, to me at least, all the models learned ImageNet pretty well, even at relatively extreme levels of dropout. They all learn the task, but how different are the inner workings of these systems? So we did next what we call representational trajectory analysis, which basically allows us to plot the similarity of all the different layers of all the different models in terms of their representational geometry. And you can see here, of course, they all start at pixel input, but by the late stages of the models, there's much more variation in the learned representations, especially around the fully connected blocks where dropout is applied. So all the models learn the task pretty well, but they're taking different routes to get there. And earlier, I mentioned that one of the benefits of low-dimensional codes is robustness. So does that pan out in this model set? We simulated the impact of damage to these models through a lesioning procedure. Intuitively, we'd expect increasing dropout to also increase the robustness of the model, meaning it can still do the task when perturbed. And so for each model, we first measured its baseline accuracy, and then randomly lesioned a proportion of units in layer ReLU6, right between the two layers where dropout was applied. We tested increments all the way from 0% to 95% of units in the layer, randomly lesioned. And then for each lesion, we reassessed accuracy, and we repeated the procedure 10 times to stabilize our measurements. So here we're plotting proportion of units lesioned on the x-axis and percentage of baseline accuracy on the y-axis. So we're normalizing all the models to have the same starting point. Looking first at the model with no dropout, as expected, we see a monotonic decrease in accuracy with greater lesion size. And also as expected, when we start ramping up dropout, robustness systematically increases. You can start lesioning many different units in the layer and not seeing basically any drop in accuracy. However, after this point of around p equals 0.7, the trend reverses, and we start seeing pretty brittle representations again when we get to larger lesion sizes. And at p equals 0.9, there's so much regularization that it becomes basically pathological for information capacity. So increased dropout confers robustness, but only up to a point. And just by varying dropout, it seems like we might have started to zero in on an optimal balancing point for AlexNet that maximizes robustness to lesions without sacrificing information capacity. Now, can these experiments tell us anything about human visual cortex? We've seen that the models all learn different things and have different degrees of robustness, but which are the most similar to human occipitotemporal cortex? We looked at data from the natural scenes dataset, examining responses in a large swath of cortex from 8 subjects. Here's data from an example subject, and these responses are highly reliable, both within and between individuals. To relate the models to OTC, we used classical representational similarity analysis. We wanted to choose a mapping procedure that was without weighting or feature reweighting or any kind of encoding models in between to test for fully emergent representational match between model and brain. And as with our dimensionality analysis, here we're looking at layer FC6. And we can see that the model that was the most robust to lesioning at 0.7 also happened to have the highest emergent alignment to brain representations. So this was interesting, and we wondered, is this correspondence being driven by having similar dimensionality between the models and the brain? So we next compared the eigenspectra between the models and the brain. And one problem that comes up right away, of course, is that the data have noise, and that noise has structure, and structure is definitely going to impact the eigenspectrum. So to deal with this, we applied a new technique developed with Kendrick Kay, which we're writing up concurrently here, called generative modeling of signal and noise. I'm happy to chat more about the details after the session, but for now I'll just say that the approach leverages stimulus repetitions to learn how to separate signal and noise, giving us basically a way to do something like denoised PCA, from which we can compute the eigenspectrum. So now we can compare brain and model eigenspectra, and here are the OTC eigenspectra estimated for 8 subjects with their corresponding slope estimates. And now looking at the dropout models, we see that the same model that was the most robust and the most brain predictive also has the rate of eigenspectrum decay that's right around the average of the human subjects, suggesting that the observed match between model and brain may be driven by a common need to balance efficiency and robustness in this representational space. So to summarize everything I've shown you so far, training identical models with increasing dropout reduces the dimensionality of their features, systematically alters their internal representational geometries while preserving task performance. And optimal robustness is observed at high but not extreme levels of dropout. And the peak of that distribution, the optimal point at p equals 0.7, also corresponds to the model that has the greatest emergent fit to brain responses and a brain-like eigenspectrum. So what can we learn from all of this more broadly? Right now, I think these networks raise more questions than they do provide answers. Is it the case that dropout is actually a plausible mechanism biologically, or is it maybe standing in for some other mechanism as a proxy? These are open questions. And it would be great if we could use empirical data to better understand the forms of regularization that govern representations in high-level vision, and understand whether the predictions of Stringer generalize all throughout the visual hierarchy in humans. And overall, I think these results really show the potential of using controlled rearing of deep nets to make sense of tensions in the literature. And even though dropout is by now a very old-school technique in computer vision, we've shown here that it provides a super easy knob on representation learning. And so perhaps rather than debating whether sensory systems benefit more from lower or higher dimensional codes, we should pursue more controlled analyses of different regularizers and models, which could help us simulate how these trade-offs happen at different levels of hierarchy for different tasks, and without requiring fancy loss functions that directly regularize the model to be similar to a brain data set or a behavioral data set. So with that, I'd like to acknowledge my collaborators and mentors, and I'm happy to take any questions. Thank you.
Thanks, Jacob. That was a great talk. We can have one question here.
Thanks for the great talk. I was curious about that plot. The one on the left corner on top.
Yes.
In Karsten paper, they showed it follows a power law distribution. I was curious if you test if yours also follows a power law distribution. And let me just add, I'm asking because it's a very characteristic distribution in biophysical system, basically.
Yeah. So we've quantified the power law alpha here, but we haven't systematically studied whether the point-by-point predictions of the Stringer work pan out in this data set yet. It's definitely an exciting direction moving forward. And not just to look at high-level vision, but also to go back to V1, because I think that would be the most direct place to do a side-by-side. Yeah.
Wonderful talk. And maybe this is kind of really related to your kind of like lesioning of the real units, but what relationship do you think you have, there is between the kind of variance in amount of dropout and the kind of like the dimensionality of the bottlenecks, right? So like you can imagine just swapping out the kind of like AlexNet layers you trained with kind of more compressed versions and seeing if there's, more, you take a hit to overall accuracy, but would that kind of increase or, the differences between the different kind of dropout levels? And secondly, if you tried something like, a beta VAE, which learns kind of like orthogonal dimensions and did drop out on those, would you expect interesting things to come out of that?
Those are great questions. So I think we'll need to chat afterwards about the bottleneck point. In terms of other models, certainly I think, autoencoders would be really interesting to look at in the context of these experiments. I'm not sure I have very immediate predictions, but I would say that, yeah, we should definitely chat afterwards. Yeah.
Great talk.
Yeah.
Let's thank Jacob again. Our next speaker is Thomas O'Connell from MIT.
All right. As humans, we effortlessly perceive the 3D structure of objects. Even with simple ones, such as these shown on the screen, we have a rich phenomenology of the global 3D shape, which generalizes even for new objects we've never seen before. So how do we do this? There are still no computational accounts of how humans generally recover 3D shape. Several Bayesian solutions have been proposed over the years, but historically they haven't scaled well beyond simple shape spaces. And over the past 10 years, the main model class that has been most rigorously tested for shape representations are deep neural networks trained on large corpuses of natural images. There's ample evidence that these standard DNNs struggle with representing the shape of objects in images. So ImageNet DNNs tend to make classification judgments based on textual information, and they show large gaps to humans in identifying objects across viewpoints. We're specifically interested in global 3D shape, meaning a viewpoint invariant representation that can be recovered from a single view of an object. Let's make this explicit with a task. We'll be using a multi-view match-to-sample task to quantify 3D shape perception in both humans and models. A sample image is shown on top, with a target image depicting the same object from a different viewpoint, and a lure image depicting a new object on the bottom. The task for humans is simply to identify the target image. The images I'm using here are rendered from the ShapeNet dataset, and they're drawn from 13 different categories. Some examples of the task are shown on the screen, and the red dot indicates the correct answer. So we ran 200 human participants on this task online. All the data I'll be showing today is plotted in the same format. So along the x-axis, you can see the accuracy on the match-to-sample task, and along the y-axis, you can see the trial-wise similarity to human behavior. So for the human data I'm about to plot, the similarity to human behavior is the cosine similarity between one participant's trial-wise responses compared to the average of all other participants. So we see that humans do pretty well on this task. They get an average accuracy of about 88%, and we have a nice distribution of responses across our participants. Next, we're going to do the same task on 25 ImageNet CNNs. So we're going to extract the activity from the last layer before the classification layer. And if the cosine similarity between the sample and target embeddings is more similar than between the sample and lure images, then we'll count that trial as correct. And we can see that the ImageNet CNNs fall completely outside the distribution of human responses here. Maybe if we use newer models, such as visual transformers, this will close this gap. So we repeat exactly the same procedure on 25 ImageNet VITs. And again, we see a very similar pattern. So some of them do a bit better, but we're still not capturing human 3D shape judgments. So what do we actually have to add to these standard DNN models to reach human-aligned 3D shape judgments? We're going to draw inspiration from a new class of deep learning models that has gained prominence in 3D graphics and computer vision. So these videos on the screen look like they might be actual videos of scenes and objects, but in reality, these are things that have been parameterized into the weights of a neural network, and when you run the neural network, you get these near photorealistic reconstructions. We're going to focus on a specific class of these 3D neural field models that can recover the full 3D geometry of an object from a single input image. So on the left side of the screen, you'll see the input image, and on the right side, you can see the 3D reconstructions from 4 of these different 3D field models. Okay, so what actually is a 3D neural field? A neural field is basically encoding some property continuously over space into the weights of a neural network. So think fields in physics like gravitational fields or magnetic fields. For these ones, the input is going to be some coordinate, so either an XYZ coordinate in space or the coordinates defining a ray through a volume. This gets run through a neural network, and the network outputs a property at that coordinate. So it could be the color or something like volume density or shape. And the network itself is just a multi-layer perceptron. So these are fully connected neural networks with nonlinear activation functions between each layer. The specific network we're using is going to encode a 3D light field. So a light field is an idealized representation of the color of every ray of light passing through every point in a scene at every possible orientation. So let's say this is the space we want to encode. We can define a camera position and an image plane, cast a ray from the camera through the volume, and then the model will take the coordinates defining that ray and provide an RGB value, which now becomes the pixel value at the point where that ray intersects the image plane. So we cast as many rays as we want pixels in our image, and after we do this over and over, we now get a reconstruction of the object from that viewpoint. If we move the camera around, this will now change our perspective on the object, and by doing that for many camera positions, that's how we produce these 3D reconstructions that I showed on the previous slide. So how do we now compute this from an individual image? We pass our image into a CNN encoder, then using a linear readout, map it to a low-dimensional 3D shape-laden space. We then use a nonlinear readout to predict the weights of the light field network MLP, and those weights now define the full 3D light field for the object. We can now apply that same rendering procedure to the estimated light field, and it now gives us a reconstruction of the object from the provided viewpoint. So 2 critical parts about this model training. One is that these models are receiving viewpoint supervision. So they are provided with ground truth camera matrices during training to tell the model what the output viewpoint should look like. And second, the loss that's used to train these models is a 3D multi-view learning objective. So the model receives a view of the object from one viewpoint and then has to render the object from a different viewpoint. And this enforces the latent space to basically learn a viewpoint invariant representation that captures the full global shape of the object. We trained a 3D LFN on about 30,000 ShapeNet objects, and the categories of these objects are the same as the ones used in the match to sample task. So the individual objects in training and testing are distinct, but the category structure does overlap. Okay, so now coming back to our data, we're going to extract the shape latents and the weights of the light field as our features to now do the same match to sample task. And notice that the rendering piece is not depicted here. This is because that's only used at training time. So at inference time, it's actually just one feed-forward pass through the model to extract these features. And we see that we've now improved markedly compared to our standard DNN models. So the weights defining the light field are within one standard deviation of both the human accuracy and the human trial-wise noise ceiling. So now we're going to push this even farther, and we're going to use our ImageNet model zoo to try and select specific match-to-sample trials that most accentuate the gap between standard models and humans. So you can see all the way on the left that the ImageNet CNNs are doing very poorly, both in their accuracy and similarity to human behavior, and as you move left to right, their performance improves. Human performance is pretty flat across all of these conditions, so this really accentuates this gap that we're trying to close here. ImageNet VITs do a little bit better than the ImageNet CNNs, but are still falling outside of human alignment. And then critically, for all of the conditions, our 3D light field models look much closer to humans than the standard ImageNet DNNs. We repeat this same procedure now using abstract generated objects that you can see on the bottom of the screen. So we retrain the light field network using 25,000 of these objects and then construct new match to sample trials using a different set of those objects. And again, we see a very similar pattern of results. So this is a harder task because there's not a category structure, so the human accuracies are slightly lower, and in the hardest condition, the light field isn't quite reaching human alignment. But it's still a clear improvement over what we're getting with our standard models. Okay, so now turning to our primary question, what's actually driving this alignment between the 3D light field network and human shape judgments? So another way to put this is, what do we have to add to standard DNN models so that they look more like humans performing these shape matching tasks? To do this, we're going to train a series of new models, modifying standard architectures so that they have components of the 3D light field network. The data shown here is the average of the 5 CNN-defined ShapeNet conditions, and all of the models I'll discuss are trained on the same ShapeNet dataset as the light field network. So if simply training on rendered objects is what's driving this effect, then we would expect all of these new models to perform equally well. So to start, the light field network is generative. It can actually visualize images of its learned latent space. So to try and mimic this, we take a standard autoencoder. So it takes an image, runs it through a CNN down to a low-dimensional space, and then reconstructs the image using a series of deconvolutional layers. But we see that this performs in the distribution of the ImageNet CNNs. So simply being generative is not sufficient to reach human alignment. Again, this is trained on the same data set, so this also tells us that just training on rendered ShapeNet objects is not sufficient. Next, we turn to viewpoint supervision. So we take the camera matrix defining the viewpoint and append that to the latent space of the autoencoder before passing it into the decoder layers. But this performs almost equally well to the autoencoder without viewpoint supervision. So it doesn't appear to be what's driving this effect. So finally, we turn to the 3D multi-view learning objective. So this is now the same autoencoder, but we've changed it so that the input and output views are different. And the camera matrix tells the model what the output view of the model should look like, much like the neural field model. And here we see a marked jump in performance. So we're now reaching the lower end of the human response distribution. We decide to push this even farther. So we take a standard ResNet 50 CNN and then use a modified MoCo contrastive loss to just give it 2 viewpoints of the same object and then try and enforce a common representation across viewpoints. And again, we see that this is performing much closer to human performance than any of the ImageNet models or the other control models that we've tested. So this tells us that a lot of the work here is being driven by this 3D multi-view learning objective. So critically, the models that look like humans see two views of an object on each training trial, and then their learning objective enforces a common representation across those viewpoints. One final note here are the different architectural features of the light field. So we do see that the light field is far and away performing better than any other model. So there could be something about the architecture increasing alignment, and we'll continue to investigate this in future work. Now, ending on a caveat, it's important to try and break your models so we now know what to target moving forward. So what we tried was to generalize models to new categories that they didn't see during training. So on the left is the ShapeNet models generalizing to the abstract ShapeGen test set. And we see that they're performing about as well as many of the ImageNet VITs, but still falling short of alignment. On the right side, we hold out one category of ShapeNet while training the models, and then construct a test set that's entirely comprised of that category. And again, we can see here that there's now a large gap between the human and model performance. So trying to close this gap and have models that can generalize in the same ways as humans is an important goal. So to summarize everything, we find that 3D light field networks in humans make similar 3D shape judgments across a variety of conditions. We find that this human alignment is driven by a 3D multi-view learning objective and actually seeing multiple viewpoints of the same objects within the training trials. We identify a limitation showing that they don't generalize to novel shape categories, and targeting such out-of-distribution generalization is going to be an important target moving forward. And then finally, we have several ongoing experiments testing the neural alignment between such 3D DNNs and recordings from human fMRI and monkey electrophysiology. So stay tuned for that soon. I'd like to thank my team of collaborators. A special shout out to Tyler Bonin for being a really integral part of this project at all stages. And that's it. Thank you.
Thanks, Thomas. I'm going to ask the next speaker to come up while you answer questions.
Okay. Just a question about the human behavior. In psychophysics, there is evidence that shape constancy is basically perfect for symmetrical objects. And as objects get asymmetrical, shape constancy gets worse and worse. Have you tested that with these kind of models?
No, we haven't looked at that. So some of the natural categories, there's some symmetry. In the abstract objects, we don't enforce symmetry at all. But yeah, that would be very interesting to look at moving forward. Thank you.
Let's thank Thomas again. Our next speaker is Clemens Bartnik from the University of Amsterdam.
Why is this? So, hi, I'm Clemens, and today I want to tell you about my research on action affordances and how they are represented in scene selective cortex and CNNs. This work is done together with Iris Huhn. So, the other day I found this video of Rambo, an orangutan, that can drive a golf cart, and I absolutely love this video. As we can see in this specific part, he's not just driving straight over the grass, but perceives his environment, takes complicated turns, and avoids objects. I'm interested in understanding the kind of representations that enable Rambo to do this and look so freaking cool doing it. So, in the past, there have been three distinct ways in which we might perceive visual scenes. This is an overview figure of our upcoming review on scene perception in the Oxford Encyclopedia of Neuroscience. One way we might perceive scenes is through the contained objects. So this is a backyard scene because it contains a play tower, chairs, and a swing. Another way might be through global properties. So fixed immovable elements like the grass on the floor or the wooden walls on the side. While these two views have received a lot of attention over the past years and a lot of research in studying these representations in the brain, comparatively little is known about how we might perceive scenes by the possibilities for actions and what they afford. This is a backyard scene because I can garden, I can play in the garden, or I can sunbathe. So in my presentation, I want to focus on this view of perceiving scenes. How does the brain represent and compute action affordances? First, I will briefly go over two studies that have looked at this. In a pioneering study, Bonner and Epstein showed that they have looked at action affordances through possible pathways through indoor environments. By representational similarity analysis, they related differences in positions of walkable paths to representations in visual brain regions to be known to be involved in scene processing, especially OPA. In a follow-up study, they showed that in CNNs, in this case AlexNet, representations in layer 5 show the highest correlations with these past representations. Another study by Groen et al. Compared different scene descriptions, including a broad set of environments, on their ability to represent similarity judgments and fMRI. Here we found that functions were important to predict behavior, but were not strongly represented in the brain and were not strongly overlapping with neural network features. But why are these results different? Although both studies compared fMRI, behavior, and CNNs, they are actually quite different in many ways. Two important factors are the types of images and the types of affordances that they sampled. To visualize this, I want to show you this T-SNE visualization of the attribute space of a large computer vision database, the Sun attribute database. And here, we can see the stimuli that Bonner and Epstein used in the representational space of scenes. They cluster together in a subspace of indoor environments. Here we can see some of these examples. They are very well suited to study affordances in the presence and direction of paths for walking in indoor environments. Here we can see the stimuli used by Groen et al. And we can already see that it way more broadly covers the representational space of these scenes. And when we look at some of these examples, we can see that these are very diverse. And in addition, Groen et al. Also used a very broad set of affordances, including affordances like socializing, transportation, and doing sports. This brings me to our current study. In our current study, the goal was to find the sweet spot between both studies. We wanted to sample a broad set of images, sampling diverse environments, but still want to focus on a narrow set of affordances by restricting ourselves to navigational affordances, similar to Bonner and Epstein. So, we ended up creating a new set of images, 231, that you can see here. When selecting these images, we took care that they don't show any people or objects, like bikes and cars, that could give away information about possible navigational affordances. In the spirit of CCN, we collected all three measurements. So we have behavioral measurements for the full set of 231 images, but I will present a subset of 90 images for which we collected fMRI data and activations of CNNs. Here, we can again see the T-SNE representation of the scene space, and we can see that our images broadly sample this space. Here are a few examples of our dataset. We sampled multiple types of environment, like indoor scenes, affording different navigational actions, like walking, driving, and swimming. Here are a few more examples, now of outdoor natural scenes, again, containing very diverse actions. And we have outdoor man-made scenes as well. Okay, now that we have this data set, we collected the behavioral measurements. For this, we restricted ourselves to 6 types of navigational affordances. In an online experiment, we let participants select one or multiple of 6 options how they would move in a given scene. Thereby, we get an affordance rating for each scene, that we can average across all participants. Like, for example, in this scene, where most participants indicated walking and biking as their options. In addition, we also collected in the same way ratings for other readily identifiable scene properties, like the contained materials, objects, or global properties. We used representational similarity analysis and computed the pairwise correlation between the mean labels of each properties. This is the resulting RDM for the navigational affordance space, and we can already see there are several distinct clusters. On the left, you see the color labels indicating what kind of environment the scene is from, indoor, outdoor natural, or outdoor man-made. In the bottom row, you can see the resulting RDMs for other properties, sorted in the same way as the action space. And we can see that these spaces overlap to some extent, but the action space is not completely explained by these spaces. Indeed, when we correlate these RDMs for different subspaces, for the different spaces, we can see that the action space only correlates around 0.3 with the others, but the other scene properties show intercorrelations of around 0.6. This suggests that navigational affordances form a distinct representational space. So this brings me to my first interim conclusion. Action affordances form a distinct representational space. So now let me turn to neural data. In an fMRI experiment, we measured both responses for 90 of our images in the 3 well-known scene selective regions. Importantly, our 12 participants saw the images under 3 different task conditions with different task instructions: a fixation cross task, a navigation affordance task, and an object labeling task. Let me show you the results. Now I will show you the RSA correlations of the 3 scene selective brain regions, averaged across all the 3 tasks, with the navigation affordances and the other scene properties. We find that navigation affordances are represented in all 3 regions. We also see correlations with the other spaces. So this is consistent with what Bonner and Epstein find, but not with what Kuhn et al. Find. This brings me to the second interim conclusion. Scene selective regions seem to represent these action affordances. But what about CNNs? First, like Bonner and Epstein, we looked at AlexNet, trained on ImageNet and Places 365. As we can see, CNNs have significant correlations with all behavioral annotations. However, only when trained on ImageNet, and the correlations with action affordances are substantially lower than with the other scene properties. Comparisons between AlexNet features and fMRI responses also show substantially lower correlations, especially in PPA and OPA. We only find positive correlations with RSC. This is more in line with what Kuhn et al. Find. Oh, so maybe a deeper network with different architecture will yield different results. So we also tested ResNet models trained on ImageNet and Places 365. But we find a similar pattern. Given recent studies showing the benefit of using models trained using contrastive learning and multimodal learning with text and images in capturing representations of high-level visual areas and also the scene areas, we also decided to test clip models with 2 different backbones. Interestingly, as you can see here, with the clip model with the ResNet 50 architecture, we see a gain in correlations with the action affordances at the expense with the other scene properties. And also that these features are represented in the scene selective regions, especially in OPA and PPA. So, we see that standard CNNs trained on scene classification do not represent navigation affordances as well, but models like CLIP might be promising models, maybe due to the fact that these representations incorporate more semantic features. Do you remember that we had different task conditions in our fMRI experiment? Previous research did not look into the task dependency of action affordances in the visual regions. Here, as a reminder, I show you again the RSA correlations for the 3 brain regions averaged across the tasks. But if we look at the correlations of these regions with the RDMs, if participants perform a color fixation task on the data, we can see that these correlations drop. Here, we see the brain and behavior correlations again when participants perform an action task. The correlations increase again. And this is for the object task, similar correlations in OPA and PPA, stronger correlations in RSC. We would expect a higher correlation with the action space over the object space, but it's quite similar for both tasks. So this might be an attention effect. So I can say that these representations seem to be task dependent. To the end, I want to give you some take-home messages. So it seems that scene-selective brain regions demonstrate a sensitivity to action affordances, representations in the 6-dimensional affordance space. Standard off-the-shelf scene and object classification trained CNNs do not strongly represent these affordances, indicating a need for different training objectives. And multimodal learning or language-supervised representation, as in CLIP, might be more suitable to capture these scene representations. And with this, I want to end and thank my supervisor Iris Kuhn and the lab. Thank you very much.
Are there any questions? Maybe in the meeting.
It's work. Great presentation. I'm surprised that you don't see any correlations with the CNNs in any of the scene regions. Do you have any idea why that is?
Again?
Did you show RSA result where you have no correlations with the CNNs in the scene regions? Did I understand that correctly? You showed RSA correlations with like OPA, PPA, and RC, and you showed different convolutional neural networks.
Yeah.
Yeah. So I'm just surprised why do you not see like those regions are usually well explained by these kinds of models.
Yeah. Yeah. We were also really surprised that we don't really find these. That we find these correlations. But yeah, we need to look further into this. What's really going on and how these are correlated.
Cool. Thanks.
More question, which is, I think another difference with prior work, I think sometimes navigational affordances also refers to the actual paths that people might take, whereas your definition of what action you might do, I wonder if that's a bit more abstract. Do you think that that's a meaningful difference?
So I think these are quite different, and we have both annotations for our data. So we have the path annotation data and this data, annotations by the different action labels. And here we want to see how different are these representations. And yeah, I would say that especially for the different actions, we need to incorporate more information than just, for example, layout information of where we can go, because different materials could give us completely different routes and also action possibilities. Thanks.
Let's thank our speaker one more time. Our final speaker of the session is Mitchell Ostro from MIT.
Thanks, Ayla. Can you guys hear me okay? Sweet. Yeah, my name is Mitchell. I'm a PhD student in the FEAT lab at MIT, and I'm going to be telling you about my project developing a new comparison method called dynamical similarity analysis. DSA is fast, easy to use, and I will present some code at the end. So let's start with an example. So here I've plotted 3 noisy samples from 3 different 2-dimensional dynamical systems, as well as their condition averages in the darker color. And they look really similar, right? So maybe it would be reasonable to say that they're doing the same thing. But in fact, they have totally different internal computations that correspond to wildly different functions that the brain performs. So this first system is called a bistable switch, which has been used to model binary decision-making and perception of this vase and phases here. The second is a line attractor that has been used to model how the brain tracks eye position. And finally, the last one is a point attractor that is used in stimulus filtering, perhaps at the level of dendrites or sensory cortex. And this example raises a problem for a wide range of different pipelines in computational neuroscience that require comparing systems. And it's not limited to this specific case. It's incredibly easy to construct systems that disentangle geometry and dynamics. Yet, state-of-the-art comparison methods are all geometric, which means that these cases are in some sense adversarial for them. So here's a list of some well-known comparison methods, and like I just highlighted, all of these only rely on the geometry of your data. And this means that if we were to use them in the previous example, we wouldn't be able to tell that the systems are different. So before I describe my solution to this problem, let's align ourselves on some terminology. I'll be taking a state space view to the neural population, which views a neural network like this one on the left in a vector space, where each axis corresponds to the firing rate of a particular neuron. And in this space, the evolution of firing rates over time corresponds to a so-called trajectory. And depending on the wiring of the circuit, this trajectory can have interesting abstract properties, such as this one depicted here, where the activity circles around one point before eventually falling onto it, called a fixed point. And these abstracted features are what we should try to compare between two systems, because these are what actually instantiate the network's computations. So, our new method, called dynamical similarity analysis, or DSA for short, can identify from data whether these two systems, or any two systems really, have the same dynamical structure, regardless of what their shapes are. And to do so, we leverage the theory of delay embeddings, which allow us to capture the full state space of a system, even when we are only able to observe a subset of dimensions, such as the Lorenz attractor here. We also leverage the theory of Keitzmann operators, which allow us to capture a global linear representation of the system's dynamics via embedding observations into a high-dimensional space. So what do I really mean mathematically by dynamical similarity? This is also referred to as topological conjugacy, and I'll provide some intuition here. So suppose we have two dynamical systems called F and G over different manifolds M and N, and suppose there is some invertible transformation phi between the two spaces. These two systems are conjugate if you can take any point S in the first system, map it to a point phi of S, or psi of S, on the other system N, evolve that point in N with G, and then map it back to M. And the resultant point is the same as if you just evolved within M via F. And this is described mathematically with the following equations. So this is the notion of equivalency that DSA can capture, but how does it work? It's actually very straightforward algorithmically. There's effectively two steps. First, we sample time series from two different systems. And here I'm depicting brains, but these are really anything that evolves over time. Then we non-linearly embed these into a higher dimension. And here we're using the delay embeddings that I was talking about earlier. Then we fit a global linear model to the data to approximate the dynamics. Finally, we compare the dynamics matrices with a novel shape metric, which I've depicted here. And this captures the notion of topological conjugacy I previously described for linear systems. So let's see this in action. Using the three systems I introduced at the beginning, we see that Procruste's analysis, when applied pairwise between sampled systems to produce a dissimilarity matrix, and then visualized here with MDS, cannot discriminate the three systems. However, DSA is able to perfectly distinguish them. Cool. Let's see the converse. We simulate a 100-dimensional ring attractor neural network, which has been experimentally identified in the brain for tracking head direction in both mice and fruit flies. After simulating, we applied a smooth, nonlinear deformation to this data, which does not change the ring topology, but increasingly modifies the geometry as the deformation parameter increases. We then compared these data at each parameter value to the smallest deformation, which is the value of 0.1 here. And we see that while Procruste's increases smoothly, as expected, DSA stays very close to 0, indicating that it can identify these systems as equivalent. Finally, we wanted to demonstrate that DSA is sensitive to topological changes, such as the tearing of a ring. So here, we applied a different transformation to our ring network, such that when the transformation parameter is less than 1, the ring is broken and is in fact a curved line attractor. And only at 1, or close to 1 due to noise, does the network actually behave like a ring. And as before, we compared the system at different parameter values to the baseline, and found that unlike Procrustes, DSA only jumps up from 0 when the parameter is close to 1. So this is, to us, strong evidence that DSA is able to capture topological conjugacy. So lastly, we wanted to demonstrate one other use case for DSA, and there's a wide variety of things you can do, but we just stuck with one other one. So in artificial neural networks, different learning rules, such as ADAM, RMS prop, etc., instantiate different dynamical systems over the network's weights and activations in response to particular stimuli. And so this is, of course, because they are based on different neural circuits, such as backpropagation and feedback alignment here, which differ based on the backwards pass matrix. And so we asked whether DSA could discriminate between learning rules based on data sampled across training. To do so, we borrowed a data set from Aran Nayebi's 2020 paper, in which he trained a variety of large-scale CNNs on different tasks with 4 different learning rules. The data set consists of statistics from the convolutional layers across training, such as the mean activation of units in that layer. And we applied DSA pairwise to sample data from networks based on learning rules, and we visualized the results here with the same MDS clustering analysis as before. The learning rules very clearly cluster well, and in fact, we were able to achieve 90% test accuracy when applying very simple logistic regression over this similarity space. And so this is in CNNs, but we're actively exploring this in neural data. So I went through this pretty fast, happy to talk more with a lot of questions, I'm sure. But the high-level takeaways I want to highlight are that dynamics and geometry are distinct features of dynamical systems, and it is dynamics that instantiate the core mechanisms of computation. DSA is a method that is able to identify these core similarities in dynamics, which we can also call topological similarities. And finally, we have code here that is very easy to use and very fast. There's a sklearn style API, and we're CUDA accelerated. So thanks to my collaborators, and I'm looking forward to your questions.
Thanks for that great talk.
Yeah.
I was curious, DSA judging purely on the dynamics of the system, not the computation the network does.
Yeah, it's the dynamics of the system. I guess you could argue that computation is something else, but that's my stance, yeah.
Super cool talk. So I'm wondering, so if I understand correctly, you basically you ask if the representation in some higher dimensional space or dynamics in this delay embedding space, there's a similarity between these two systems. So can you assign some interpretation to that matrix? By which I mean, can you sort of get back to the original coordinate space and ask if there's an explicit representation of the dynamics for those two systems that's shared?
Yeah, so there is a direct mapping from the delay embedding space to the original space. You just read out the top dimensions, if that's what you're asking.
Yeah, so I guess can you recover the actual explicit equation for the dynamical system?
I would say probably not, no.
Okay. Thanks.
There also might be a wide number of equations that correspond to the same system.
Gotcha.
Hi there, thank you for your very cool talk. I was just wondering, one part of the step that you mentioned had to do with coming up with like these best fitting linear models for dynamical systems that you're comparing with one another. And I was wondering if there are situations where you're potentially dealing with maybe higher dimensional attractors where you actually need to come up with sort of higher order fitting models and then compare those rather than just looking at best fitting linear models.
Yeah, so there's a wide number of embedding methods that you could potentially use. Here we just chose delay embeddings because it was efficient computationally. But yeah, no, this is able to work for nonlinear systems very well. And people, you might be asking, oh, okay, you're approximating a linear system, or approximating a nonlinear system with a linear one, that's not fair. It actually doesn't matter in this context. There's some really cool theoretical results that we have. Even if you don't perfectly approximate the system, you can still identify this type of conjugacy.
Thank you.
All right, let's thank Mitchell and all of our speakers from the session. I'm going to tell. All right, so please come, if you're coming in just now, please come find your seat quickly so that we can get started with the keynote talk.
All right. While the last few are filtering in, I just wanted to make a quick announcement about the social event, which is going to happen right after the final keynote for today. So if any of you, oh, okay. Can you hear me okay now? So the social event starts at 8, and you're welcome to make your way there on your own if you like, but there will also be boats leaving there at 7:30 and at 8 and at 8:30. They'll be leaving from Folly Bridge, which is about a 15-minute walk away from here. Again, you can go there on your own if you like, but if you're not quite sure how to get there and you'd like to go as a group, we will be leaving as a group from the front entrance of the exam schools at 7:15 to get the first boat, or at 7:25 to get the second boat because the exam schools close at 7:30. Remember to bring your ticket and your drink tokens, because you'll need those to get into the venue and onto the boats. And I'll hand over to Thomas now.
Thanks, everyone. So it's my pleasure to introduce Tim Keitzmann, who is the last speaker of the day and who is very excited about getting on a boat. End of his talk, as am I. So Tim received his PhD in cognitive science at the University of Osnabruck. And after a postdoc at Cambridge, he became an assistant professor at Donders and is now a full professor at the University of Osnabruck. So Tim's work focuses on the relationship broadly between AI systems and the brain. And as I think you'll now see, he's made some really impressive progress using new developments in AI to gain insights into human vision. So go ahead, Tim.
Thank you very much. Thank you. So, first of all, thank you for showing up after a long day of conferencing. I'm very impressed with the number of people. To the people on the right, as a quick disclaimer, I have bad news for you. I screwed up my neck, and looking to the right is not one of the things I can do at the moment. So it's not that I don't like you, it's just I won't look at you. First of all, thanks to the organizers of this conference. It really is my absolute favorite conference, and I know that's a thing people say, but I do really mean it, and it's an absolute honor to give the keynote today. And with that said, I noticed in preparing the talk that I was nervous, and I was like, no, I usually talk to like larger crowds, and I'm not nervous. Why am I nervous for this particular occasion? And the reason is that I care very deeply about this community, and all people whose work I adore, many of them are here. So thanks for making me a bit nervous. You're a great community. So maybe we'll start with a few words on the lab. We're currently 7 PhDs and 3 postdocs. There's 2 things that we obsess about as a great deal. One is coffee, and the other one is research. And when we don't obsess over coffee, we wonder about the neural computations underlying primate vision. And we operate in the space of neuro AI by using techniques from AI to make progress in understanding neuroscience and trying to embed neuroscientific insight into AI to hopefully at some point, it's a marathon, not a sprint, improve ANN models in application by taking inspiration from neuroscience. So in German, there's a saying, you can't dance at 2 weddings, but I think that neuro AI wants to do exactly that, right? So that's what we're trying to do in the lab as well. We're trying to dance at the neuroscience wedding, and we try to dance at the machine learning wedding. And one element as to why we can do that is because we use ANNs in our day-to-day research. And there are, in sort of our hands or the way we use them, they're like a common language that we can use to create computational models of neural computation, while at the same time having models that do something, that perform tasks on visual input. Okay. A lot can be said, I guess, about using ANNs as models of biological vision, and that's the main focus of the talk today. In fact, so much can be said that we got together with a large group of people and wrote a paper about it. This paper, I'm showing it here because, first of all, because it took us years to get there, because we had to sort out our thoughts on why we are so excited about using ANNs in this space of computational neuroscience. And so we've basically put it all in there, why we think ANNs are cool, why we think they're useful, we ground them in the philosophy of science, introduce the tools, the historic trajectory, 300 citations to all of your work, basically, it's all there. So do check it out. I think it's if you're not familiar with this space, it's a good introduction. And I want to start the talk by, and you can see that's a huge group endeavor, obviously, and all projects that I'm talking about today, three of them in total, will be collaborations among 3 or more labs that got together to push forward. And I think that's something that is very unique maybe to CCN, that people are very, very collaboration friendly and eager. And it's not like just individual labs doing their thing, but whenever you have some problem, people are happy to jump on and help. So I'm going to highlight some of the. There you go. I'm going to highlight some of the aspects of the paper first, which are a bit more theoretical. It's probably absolutely clear to most of you, but to the ones that are not, we think that ANNs are useful because they live in a Goldilocks zone of biological abstraction, where they strike the balance between biological realism and algorithmic clarity. So what is it that I mean with that? So think of astrophysics. In astrophysics, there's the Goldilocks zone, right? So if you're too close to the sun, it's too hot and there's no water, so there cannot be any life. If you're too far away from the sun, it's too cold, there's no water, there's no life. But there's the Goldilocks zone in between, which is not too hot and not too cold, and there people can live and thrive, and we think of the space of a distance to the brain as to where models can live and thrive. So think of the space of biological realism, and if you're basically replicating a brain in silico, then you're all the way up there in the red. And Grace put it very well in Grace Lindsay in her paper, Models of the Mind. A map the size of the city has no good use. So for us, the essence of modeling lies in an act of abstraction, abstracting away from the system that you're studying so that you can start understanding what individual elements of the system are doing, and that you have a simpler mechanism that you can actually understand rather than just replicating what's there. And so we think that ANNs operate at this productive level of abstraction where they're potentially realizable, and we can argue about that over a pint later. But they're not too detailed, which allows for clarity and computational tractability. I think that's very important. So the units are very simple, but we have many of them. And so that means we can train them, we can backprop on them, and we can make them do tasks on images. And that is 2 features that I deem useful in modeling vision. Specifically, that's my home turf. That I would like to have models that I can compute on images, and they go all the way up to behaving in some way or another, and I can look at the individual computations that are in between, and ANNs allow us to do that. So given that we can train them and we can push a lot of domain knowledge into them, we can have this, we call it Neuroconnectionism research cycle, where ANNs, as you've seen probably 10 times at this point at the conference, are defined by the architecture, training set, loss, and learning rules. And basically, to us, that is just a handle on testing hypotheses about the brain. So really, you're asking, hey, what if I change this connectivity from feed forward to recurrent, or what if I change this and that? Having a hypothesis about what sort of elements might underlie a cognitive function that you would like to study. And then you bake that in, you train the model, and then you test it against brain and behavioral data across many scales. And so that is, to me, a very productive way of doing this, because it allows us for expressing and testing hypotheses about brain computations in these image-computable and task-performing models. Now, the next slide is a bit awkward, because I always have Jay in the slide, I met him in Cambridge once, and I was so starstruck by the clarity of thought that I started putting him into my slide as an homage, and I thought, why stop now? When I started working in computer vision, so the slide is called End-to-End Training and the Limits of Human Creativity. The idea is, when I started in computer vision, the way we approached problems was basically look at the problem. Let's say you want to classify fruit, and then you would sit down and think, okay, what features are useful? And you would think of, color might be good, major axis rotation, shape features, you program them up, run an SVM on it, and get some performance. And really, we were a bit limited by our own creativity as to what features we came up with. And deep nets sort of solve that issue. They move our creativity to another level, if you want. They solve it because they're trained end-to-end, which means they tell us what features are sort of sensible given the task setting and architecture that you operate on. Sensible. So now we don't have to worry about the exact features anymore. We don't design them anymore, right? Feature design is not a thing anymore, but we worry about higher level sort of ingredients. And sometimes, and don't kill me for this, but sometimes I'm a bit reminded of the state in cognitive neuroscience a few years back, where we would have experiments and we would think, faces, well, you can say I grew up in vision because that's the examples that come to mind. Let's do faces and houses and bodies and maybe tools, and contrast them and look at what sort of brain regions light up. But fundamentally, in the sort of complicated features that the brain may use, especially in intermediate vision, we were a bit limited in the sort of experimental designs that we did. And ANNs give us a handle on these complex features that now we can use them. We can then run encoding models and RSA on them and compare them to the brain. And those features we could have never come up with or designed, right? So that, I think, is incredibly powerful in thinking about brain computations. Now, when I say this, people typically respond, no, not typically anymore, but that used to be a typical response, that these models are so parameter rich, like how can you possibly make sense of them? And it's all just curve fitting. And I'd just like to remind you that the experimental logic is very much the same as it always was, right? So usually you had a hypothesis about brain function, you came up with your experiment, faces and houses, you contrasted them, and you got some brain images out, and you would say, okay, does OFA or FFA care about this sort of experimental paradigm? Now, it's exactly the same thing with ANNs. It's just getting to the actual model is a bit more complicated, but really you bake your hypothesis into the design of the model, you train it, and then you compare the trained models to your brain data, and you allow to make inference about the high-level ingredients of the model, the architecture, the training set, all these things, by making these comparisons, right? So we don't actually worry about the many millions of parameters in the model, because we worry about the sort of higher level number of parameters, which are much lower than the number of parameters in a given model. Now, there's a bit of ANNs polarized, let's just say. Sometimes there's a paper coming out saying, hey, this ANN, we tested it in this experimental paradigm and it sucks. And so all ANNs suck. And so that's part of the reason why we wrote this overall paper, that we thought really they're a language for thinking about computations in the brain and implementing them. So in each individual sort of instance of a model, if that doesn't work on something, that's useful, but it's not disqualifying the whole field. And so in the paper we argue, taking inspiration from Lakatos and the philosophy of science, that every sort of research program has a central core that people buy into, that they say, I think this is a useful way of thinking about brain computations. In our case, that would be distributed connectionist systems. And then you go out and you test hypotheses about this sort of core belief that you have. And it's really not about whether an individual instance of that core is like the perfect one model, which we will probably never have, but it's really about whether we as a whole field have make progress over the years, right? So there's this distinction between the core and the belt in Lakatos, where the belt are instantiations of your core that you go out and test. And as long as there's movement in that belt, you're fine. As long as you add new knowledge. Your research agenda becomes stale if you don't add new stuff to it. So failing on something is not bad, right? We call it a signpost in the paper, right? If there's no fundamental reason why your core is wrong, you can be fine. So we argue for a more longitudinal view of using ANNs, let's say. So with that theoretical bit out of the way, let's look at two instances of where we've recently sort of added things to the belt, I think. You tell me later. and that I would like to highlight. One is a preprint that's about a week old. That's also a reason why I couldn't send Leila the abstract for my talk for a long time, because that was still in the making and I really wanted to have it in here, and I wasn't sure whether we could. Okay, so the one project, it's a preprint that's out now. It's called End-to-End Topographic Networks as Models of Cortical Map Formation and Human Visual Behavior Moving Beyond Convolutions. So here we dealt with the emergence or the study of topographies, and I think that topographies are a particularly interesting research area because topographies emerge over various timescales, right? Evolutionary, over the lifetime, and so experimental manipulation of these things is hard, also ethically questionable, but also hard, just from a toolkit sort of standpoint. So there's a special role for computational models in this field of studying topographies because they allow us to manipulate all of the various factors that there are that may contribute to various topographies that we might see out there. So they're really like a solution, to me at least, they're a solution to this conundrum of studying topographies. We bake them into models and we see how they behave over time. Again, model contrasts will be quite important. So this is work with, I should add, this is work again with Zijin Liu, Adrian Durick, Vicky Bosch, Bas Kramer, Dan Kaiser, and Radek Zishi. So it's 3 different labs who came together. So us and Daniel and Radek came together to sort of see how far we can push these things, and where we approach this from the modeling side here. Obviously, I don't need to tell you that there's topographies in the brain. There's across various different levels. There's, orientation columns on early visual cortex. There's beautiful work by Talia on the tripartite organization of the visual system. And of course, we've seen plenty of macaque face patch data so far from Freiwald and Zhao and other labs. Now, when you look at brain score and these models that we typically use to predict visual cortex data, many of the models are convolutional, right? And so convolutions fundamentally are at odds with topographies because by definition you take the same feature all over the place, and now if you look to the left you see that that's not the case in the brain, right? So there's another class of models that study topographies, and those are smaller scale models that are not trained end to end, right? They're like proof of principle sort of models, and they're super useful, don't get me wrong. But what we wanted to do here is to sort of strike a balance between these two things. We wanted large scale end to end models that would do stuff and that are image computable, but at the same time models that would have a topographic organization, right? A true topographic organization. So there's 3 desiderata that we wanted our model to have. One is locality, so a feature at a given location should be learned locally. You wouldn't, so the hashtag of the project that we had internally was the brain doesn't copy paste. So if you learn something in one location, you can't, like a convolutional kernel, just copy that knowledge all over the place. You need to learn locally, otherwise it's not a good model. You would like the units to be arranged along the cortical sheet, ideally, and you would like the features to be smooth. Smooth, why? Because we know if you look at that data, it's not salt and pepper. There's some autocorrelation, spatial autocorrelation to the feature selectivity in the cortex, so that's something that we baked into the model. So what have we come up with? This is a model that we call all topographic neural networks. They're called all TNNs, an homage to the all CNNs that many of you may know. The first network that was like just convolutions stacked on one another. These are just topographic elements stacked up on top of each other. Each unit in each layer of the model belongs to a hypercolumn. A hypercolumn has the input from, all units in a hypercolumn have the input from the same visual location. And as you move about hypercolumns, you move across visual space. And each layer has this sort of topographic arrangement as you go up. It's a 6-layer network here. And again, they're end-to-end topographic. So it's not that we have a convolutional backbone that would do some heavy lifting for us in the encoding, and then we move the units around for topography. This is supposed to be like full-on a topographic sort of organization here. And they're image computable and task performing, so we can use deep learning on them. So now let's train them and look at what happens. And there's a quick shout out to another project that we've completed a while ago when I worked with Nico, and this is EcoSet. So if you haven't heard of it, and if you're annoyed about all the dogs in ImageNet, this might be your solution. EcoSet is a large-scale vision data set, 565 basic level categories. We took them as the most frequent nouns that people use in everyday speech and like large language corpora, and it's object categories that are concrete, so stuff you can visualize, like tables and chairs, and not democracy or love, right, which is hard to visualize. So 565, 1.5 million images that you can play with. It's freely available. So dog is just one category, not 120 in this. Right. Okay, so here's one of those layers from the ALTNN to the top left. This is before training. You can see there's no smoothness sort of baked in from the get-go. But oh, sorry, I missed an important bit. How do we train the models? So there's two objectives that we're trying to fulfill at the same time, and that's super important. I'm sorry that I glanced over that. The models are trained to categorize visual input, usual cross-entropy on this ecoset dataset that I showed you, and at the same time we have a spatial similarity loss, which means that we take nearby units and we take the cosine and the weight kernels, and that should be, so the cosine distance should be low, which means the model tries to be as smooth as possible while performing the task as well as possible, right? It's a trade-off between these two things. Think of the spatial smoothness as a sort of regularizer, if you want, right? Okay, good, that was important. Okay, so this is a plot. This is the first layer, and we color code different orientations. So you train the model on this task, and then you show Gabor ratings of different orientations, and you just highlight which unit is most selective to which of the orientations. And we will go through the training as a little movie in a second. To the right, you see the performance go up. The performance, disclaimer, is nowhere near what you get with convolutional networks. They're an engineering hack, and they're a brilliant engineering hack, right? So don't expect this to be like super high performance. That's not our task anyways. Okay, so let's go through this. I hope you can see that the topographies start changing, and at about, let's say, 40, 50 epochs or so, you get a relatively stable topographic arrangement. That then stays stable while the performance of the model keeps increasing, right? So you get this early on, fairly early during training, you get like a scaffolding of topography that then the model keeps optimizing to sort of optimize this performance. And once it's trained, you get this sort of map. And there's multiple things to be said about this map. First of all, it originates fairly early on. Second of all, there's areas of shared orientation tuning, as you may remember from early visual cortex, right? Nearby units are selective for similar orientations. And one thing that we didn't quite expect, but in hindsight is 10 out of 10, that we didn't quite expect was that if you think of the feature variety, if you look at it towards the center of the image, there's much more varied features in the center than in the periphery. Right? There's larger areas of homogeneous selectivity in the periphery compared to the fovea. And why is that? And it's very likely that there's a photographer's bias in ecoset because objects are in the center. So the model learned to trade off the both objectives, not globally across the whole network, but locally. So you can afford similar feature selectivity in the periphery, but in the fovea, you really need all the diversity to do the classification task, so you pay in smoothness in the fovea so that you can do the task well, right? And so what happens is if you drop out units in the larger regions where there's a lot of redundancy, if you want, that doesn't really hurt performance much, but if you drop out units in this foveal area, there's much more drop in performance, which means that the model can pick up on these dataset statistics and make use of them and lead to something that reminds us a little bit of cortical magnification, if you want, right? Okay, if you were to not enforce smoothness, that we call LCN down here, that is basically the same model. You just set the alpha to 0 for the spatial smoothness constraint, right? Then it doesn't give you any smoothness. So it's not the dataset, not the autocorrelation in the dataset statistic that gives you smoothness. We need to actually enforce it. And convolutional networks don't have it because why would they? Because they don't have topography in that sense, right? Now we can go to the late layer of the model. And the same thing happens. After 50 or so epochs, you see a fairly stable scaffolding of selectivity. Here what we do is, again, classic cognitive neuroscience approach by having scene spaces and tools. So we run an in silico experiment on the model, and this is the D primes for these three categories, and you see that it does emerge with areas that are phase selective or tool or scene selective. And again, that's nothing you would get for free, so you need to sort of push the smoothness into the model by this objective. And I'm not happy about that, but I'll talk about this later. And convolutional networks, again, don't do it. So clusters of similar category selectivity emerge in later network layers. And it's all trained at once, right? So all emerges at the same time. It's not that we would train one level and then freeze it and go on and on and on. No, it's like the whole thing trains at once. Now, what we wanted to know was, we were intrigued by these findings, and we thought, is that model, can it pick up on spatial regularities that you see in everyday life, and can that be mirrored in its behavior as much as you can see humans sort of showing positional dependent effects of categorization, right? So that's where the other teams come in. We have a 5x5 grid where we showed the object instances of 16 categories in all those locations. We blip it on for like 40 milliseconds and then mask it, because our models are feed-forward, mind you, and we try to kind of keep people also on their feed-forward paths as much as we can. And so what you get is accuracy maps for humans, and you can run, right, for every location, how good are you at this location for this category that we were testing, right, and we've got 16. Now you can do the same thing using, let's say, the test set of your training and show objects, or you can take all of COCO if you want, and show objects to the networks, and you can get accuracy maps for the networks as well, right, for convolutional nets and for the topographic networks that we studied. So the first thing, so now we've got a whole lot of accuracy maps and we need to make sense of them, right? So the first thing we asked was, okay, are the locations where objects occur more frequently in COCO telling us anything about how good people are at those locations, and how about models? So we take, very crude, take all the occurrences of objects of a given category in COCO and create a frequency map of the same size as the accuracy map that we have, and then you just correlate them. Right? And what you see is that indeed there is locations where objects occur more frequently are locations where people are a bit better, and the all-TNNs follow that, I would argue, a bit more closely than convolutional networks that don't show strong positionally dependence. Right? So next what we asked, so that was nice, but we wanted to know which of the models aligns better with the human data more directly, right, other than going this route across image statistics and frequencies. So the next thing we did was we took the accuracy maps of humans and the all-TNNs and humans and CNNs, and we correlated them directly. And here we found that the all-TNNs correlated more strongly with the human accuracy maps than the CNNs again, showing us that whatever they pick up is something that sort of mirrors human behavior. Now, one thing that I've shown you before is that there's a central bias in the model, right? This fovea magnification sort of reminiscent feature. And could it be that this is all just central bias, right? People are good in the bang on in the center, and if all TNNs do that as well, is there anything that's, and that's, I mean, if only that were true, I'd also be happy. But is it object specific as to where these models perform well? So is it just a central bias, or is it object specific? So for that, we basically borrowed representational similarity analysis, but instead of patterns of activation, we put the accuracy maps in. Now you get a whole matrix that tells you how similar all the accuracy maps are to one another. And now we can compare those patterns of accuracy map similarities across, again, humans and all TNNs and humans and CNNs. And the idea would be, if you have a unique central bias that doesn't care about the object category, that would be a constant, and if you correlate the two maps, the constant is typically taken out, so that should clear that right up. And here again, we see that the all TNNs outperform the convolutional networks, telling us that maybe to those patterns that we observe, there's some object specificity. The network picks up some regularity in the world that humans may also latch onto, right? So as a summary of the first project, and this again is a first step in a probably long sequence of experiments that we'll run with this. Is we're just super excited to make them work first of all. So as a summary, all TNNs are an architecture that enables us to study topographies and end-to-end topographic image computable and task-performing models. It's a bit of a mouthful, but it summarizes nicely all my desiderata from what I wanted from neuroconnectionist models. They're trained on, when you train them on a meaningful visual diet ecoset, I mean, it probably works on ImageNet as well. They exhibit topographic features that remind us a bit of early visual and higher level organization. and they outperform convolutional networks in mapping onto human behavioral pattern if you look at behavioral patterns over space, right? Note, and that's what bothers me, and if you have a solution, come talk to me. What bothers me so far is that we have to have the spatial smoothness constraint baked in there, right? So we have to say nearby kernels need to be identical so we get the similar autocorrelation as what we expect from the brain, but I suspect that we don't grow up by having, you know, an external sort of smoothness constraint. There must be something else that makes, and we have some ideas, and there's ongoing work. If you have ideas, approach me. I'll be curious to get rid of that constraint and have it emerge naturally from the data. Okay, that was project 2. Well, Belt project 1. So Belt project 2, you may have seen that we changed the architecture, and that's one thing that I really appreciate about people using ANNs in computational neuroscience, is that every single aspect, so let's say we started with AlexNet, and ever since, every single aspect of AlexNet has been taken apart, changed, retrained, tested, and so we looked at the effect of input of visual diet by introducing EcoSet. We've looked at the effects of individual differences. We've put recurrence into these models. Now we start looking at topographies, and many others obviously have done all of these things as well. And so the next project, we ask about the objective. And this is, again, a huge collaborative effort, spearheaded by Adrian Durick and Jan Charist. It's Adrian Durick, Emily Allen, Jan Wu, Thomas Nazalaris, Kendrick Kay, and Jan, who came together to work on this. If you've seen this preprint before, it's September 22, it's almost a year ago. We've done a whole ton of work since then. It's not yet in the preprint, but I'll show you all the new stuff now. And it's one of these cases where reviewer feedback was just so valuable. And so if you're here, thanks. Okay, so let's ask about the objective. So let's think about the textbook view of what the visual system does, or what the ventral stream specifically does. You know, if you open a textbook, it will tell you the ventral stream is the what pathway, right? It tells you what something is. It tells you that it's here to categorize stuff, for instance, right? And that mirrors nicely in, like, including my experimental work. Back in the day, you show sequences of objects that belong to different categories, and it's random sequences, central fixation, and then you contrast these object categories. You may vary, you know, the viewpoint, the size, all these things, and show that you can decode object category from ventral stream. And that got us to a point where, you know, we have this view of the ventral stream as, you know, increasing receptive field sizes as you go from early to later visual areas, increasing complexity, and you go from, you know, edges to like texture-esque features to, you know, a category-based code, or a code that allows you to read out categories very well. And interestingly, and maybe, you know, interestingly mirroring our experimental approach of showing categories to people in these random sequences, that's how we train deep nets, right? It's random sequences of objects that the networks are trained to categorize. And so that also, you know, a lot of the initial progress with, you know, AlexNet and BGG and all these things were based on this view that object categorization is a good proxy for understanding what the ventral stream does. And I'm very aware, I look at Talia, I'm very aware that, you know, there's a ton of unsupervised objectives now. But I think, like, if we thought of a, you know, textbook sort of view, then it would be like what and categories. So what we thought in this project was to think about, are categories the final stage? Is that, like, can we get a level more? Can we get more complex as a feature description of what the ventral stream might be doing, or what the visual system might be doing? And I think it is summarized quite well in this picture. If you look at this picture, and I told you laptop, cat, and mug, I hope you would agree that the complexity and richness of this visual scene, you know, cats doing what cats do, is not really captured by just a bag of category labels, right? Saying, you know, laptop, flowers, desk, mug, phone, right? So we really wanted to get at, you know, can we get closer to like the semantic understanding of a scene, or the complexity, the richness of a scene? And that's hard. Jan's insight was, hey, let's just take the natural scenes data set, and each scene is a complex natural scene, and each scene is sampled from COCO, which means that each scene comes with 5 captions. And now the captions give you more of that richness, right? So the caption would be a cat stretching out on a laptop on a desk or something, right? That would be more, or transporting more of the meaning of the scene than just the labels. So if that's what you want, then you need to define a model that can give you a good proxy onto image captions that you can then use and test against the brain data, right? And so that's where large language models come in, or language models at large come in. And we thought, let's just try that. Let us take data where people were in the scanner, they see an image, and we collect fMRI data. And then on the other end, let's take that image, get the caption, and throw that caption into an ANN, a language-based ANN, right? Let's say an LLM or so. And maybe we try to model visual responses using language models, and maybe that works, because it's a more complex, richer sort of representation. But it felt very wild at the time. And so let's not just jump straight to GPT-4. Let's go step by step. So our first model that we want to use is a multi-hot bag of categories model, which says just a multi-hot model. It has ones where all the categories of a given scene are and zeros otherwise. We call that like a category-based code. And then you can take the nouns of the embedding and you can put them into fast text as like one step up from, you know, going into linguistics, let's say. You can do that with verbs of the caption. You can take all words from the caption and put them all in and get them all into fast text, get the average fast text embedding. And then you can go and say, okay, let's take the whole sentence and take a newer model, the Google Universal Sentence Encoder, which is still a bag of words model, right? The version we use. It doesn't care about syntax or word order. And put it into GUIs, or let's download like the latest semantically trained LLM, which is MPNet that we used, and put the captions into that. And so really what we get from all of these models is a high-dimensional embedding of the captions in a language model, right? And then you can use that to run RSA like you always do, right? And just don't worry about that the people have seen images and now you're using language models to sort of model that data. Okay, encoding models work as well. Rest assured, it's not RSA specific, but I'll show you the RSA stuff. So now let's look at ROIs. We have an early visual cortex, and then we go across the three streams, ventral, medial, and parietal. And let's start with the ventral stream and the higher level ventral stream ROI, because that's the one that I think most textbook-ish is the classification sort of area. And let me see my cursor. Well, aha, okay. I forgot I can't look to the right. Okay, I'll just tell you. So if you look at the category multi-hot to the left, you get about like 0.2 correlation of the RDMs, and this is using all NSD images per participant, so about 10,000 or so images per participant. So it's a really rich, huge sort of space that we're trying to capture here. It's not only a small fraction, it's all images. And then if you go to category word embeddings using FastTech, that doesn't really do much. Verbs are not a good idea. If you put all the words of the caption and take the average fast text embedding, that's the fourth bar, all word embeddings, that's a bit better. But it turns out you can do even better if you go to full sentence captions embeddings, as for instance GUIs or MPNet. So the jump is from, let's say, 0.2 to 0.3 something in correlation. So quite a substantial jump, I may say, because if I look at our usual manipulations of ANN architectures, the effects are much smaller than that. So let's look at all the other regions. What surprised us was that we could see a similar pattern across all of the visual regions. There's a clear transition so that language embeddings are overall better for later visual areas. So you can see that the green is a bit lower than the yellow on average. But it is true of all of the regions that the models that would give you a whole sentence embedding, GUIs and MPNet, would give the best sort of mapping to all of those visual regions that we've looked at. You can perform a searchlight on this as well, and you can see that these language sort of embeddings explain variance all over the place, right? Now, there's a problem here, and the problem is that the language models that we use were trained on whatever, right? MPNet is like this huge trillion corpus. We'll take whatever we find and train something on it. And we vary all of these things at once, right? So we change the objective, we change the architecture, we change the data sets, it's a nightmare. And so what I tried to make clear in the start of the theoretical bit, that model contrasting, careful model contrasting, is what's incredibly important to sort of getting insights into these models, and that maybe is a bit different for neuroconnectionism compared to many AI applications where whatever works, works. And so here we need to contrast these things and be very careful about controlling as much as we can. So onto ANNs. What we've done is take a recurrent convolutional network, a BLT model, and we've trained based on images. So you take all of COCO, you put the images in, and your task is to predict the language model embedding from the image. So you get like a transition in the model from a pixel-based code to a more and more high-level linguistic sort of code. I hope the linguists in the room don't kill me for it. But let's say there is a sort of like trajectory from vision to more high-level semantic features, and you have these hybrid features in between. And now we can train this model on a multi-hot code, again, a category-based code, tell me what the categories are in the scene, or we can train it on this sort of semantic embedding, right? And now we can contrast these two models, and that is incredibly important because we've now fixed it all, right? They see the exact same images, it's the exact same architecture, it's the same learning algorithm. The only thing we change is what they target at, what they're trying to predict, right? And that's important because we've seen that some groups are also using CLIP, for instance, as a model of ventral stream. And with NSD and CLIP, there's this caveat to be had that CLIP is trained on the COCO images that are also NSD. So the model has seen during training the very images that you now use for testing. And you can cross-validate it, but it makes me feel a bit nervous. So here we took out all of the images that are in NSD, and we trained only on the rest of COCO to make sure that we wouldn't do that mistake. Okay, now we'll again have 4 models, 2 of which I've shown you before. The multi-hot bag of categories is again this multi-hot code that we had in the slides before, and the LLM and PNet sentence embeddings are literally the sentence embeddings that we target on. And now we have the 2 models that are trained on the 2 things, on the multi-hot and on the LLM embeddings. And now we're going to compare it again against ROIs. And to the very left, you see the multi-hot code. So that is just data I've shown you before. A multi-hot code works better the higher you go in the ventral stream or the higher you go in the visual system more broadly. And the multi-hot trained RNN is better than its target, right? So that's nice. The taking visual properties into account in the model gives you an explanatory advantage over the target that the model is trained on, the multi-hot code. What gives you an additional advantage again is the MPNet embedding, even better than the RNN trained on categories, right? But if we train an RNN to predict the LLM embeddings, you get yet another boost. And that boost is now above the language model that I showed you before, right? Which is neat. So the image computable model had something in addition, in terms of explained variance, in addition to what it's actually trained to do, right? Because it can't fully do this task. It's not perfect. There will be some visual elements in the model still. And those, in fact, help the model explain ventral stream computations. I should have said, this is the last layer and last time step of the recurrent model that we use here. You know, the most abstract thing that we could get. Now you can do model contrasting. Again, you can contrast the model that's trained on the semantic and the category objective, and you see that it's pretty far spread significantly better than the model trained on the multi-hot, right? So it's 2 image computable deep nets, just one is trained on this semantic embedding. And again, we control the visual diet, architecture, all these things. So I hope that I convinced you that we demonstrate here a clear progression in predicting brain activity as models incorporate increasing levels of semantic information, from just multi-hot codes of categories to fast text embeddings that are just co-occurrences of nearby words, right? To GUEs, which aims to, extract the whole semantics of a whole scene, of a whole sentence, versus MPNet that's specifically trained to do semantics or semantic understanding of language. This is observed across the entire visual cortex. That was a bit of a surprise to us. It replicates in the models. It replicates with various different measures, encoding models and RSA. So I'm very certain it's legit. But it's not only the ventral stream. It's the ventral and the dorsal stream as well. So it's not restricted to a small number of just like semantic areas. So we are careful, and we would like to say that this warrants a change in perspective. Nevertheless, the visual system seems better modeled as a system aiming to transform visual inputs into a semantic scene information than aiming to transform input into object inventories, right? And this is not to say that the brain does MPNet, right? That's an important thing. We just think that the features that we get from MPNet are richer and more semantically viable, and they lend themselves to sort of explaining more variance. And we still need to figure out how to raise models so that these sort of target features emerge from themselves. And that clearly, and that connects to Jay's talk, that clearly warrants that you need some multimodal sort of raising of these models with visual diets and being spoken to and so on and so forth. Okay, the current best models that we observe are these recurrent convolutional neural networks trained to predict semantic embeddings. I haven't talked about the recurrence bit. I usually love talking about recurrence, but today I skipped that. But the recurrence bit gives some neat results in showing that, you know, later responses I'll make sure not to screw it up now. I think later responses of the early layers better predict early visual cortex, and early responses in higher level representations predict early visual cortex better. So we think that Adrian is nodding. Good. Okay. So it sort of explains that, or it suggests that what we're measuring in the fMRI data of early visual cortex is also potentially, let's be careful, but re-entrant information from higher level cortex. And we get at that temporal resolution not because fMRI gave it to us, because fMRI is temporally sluggish, but because the model gave us this temporal dimension that we could harness to understand the computations in the brain. Okay, time for a general summary. ANNs live in a Goldilocks zone, not too hot and not too cold, of biological abstraction, striking this balance between biological realism and algorithmic clarity. We're not done by any means. It's important. When I say they're the current best models, I don't mean that we're done at all. It's just I'm excited about the tools that we have to sort of ask new questions. Neuroconnectionism isn't downloading AI engineering models and running them on brain data. It's very careful about contrasting models and training them and making sure that you've really gotten the differences between the models down. And we've seen two examples here where we've updated the training objective and we've updated the architectural design, once in topographies, I had that the wrong way around, once in topographies and once in predicting semantics. And I hope that I convince you that neuroconnectionism as such is a very progressive research program. One thing I want to say before I'm done, for people being annoyed that ANNs don't yet do what they are supposed to do, that's fine, right? Be annoyed and show us where they don't work, because this is the biggest service to the community that these people can do, to show us where they don't work, because we like seeing where they work, and sometimes we're blind to seeing where they don't work, so show us where they work. And for people working with ANNs to explain stuff, you know, we need to be thankful for this criticism, right? It's a huge service to show us where this stuff doesn't work. But let us also show where it works, because that's cool too. Okay, with that, I thank all lab members for the tremendous efforts that went into, and all the labs that have been involved. Again, it's just me presenting the work of many labs having come together. Special shout out to, you may have noticed that there was one name occurring from like project 1 to 2 to 3, and that's Adrian Durick. He's great, he's in the bottom left. If he approaches you for a faculty position, don't think twice and see me cry for a week, because he will leave my lab and I will be very sad. Anyway, thank you very much for your attention. I'm looking forward to your questions.
All right, thanks. We have time for talks, so we'll go counterclockwise. Let's start there.
Thanks for the wonderful talk. I had a question about the models that were trained with topography in mind. I was wondering if you considered or looked into the kinds of like adversarial images that these models might be subject to. Things like, you know, like CNNs typically are very sensitive to small perturbations and noise, where like an image of a panda will suddenly, like with a little bit of noise, will turn into a pancake. And of course, the visual system is also subject to these metamers where, you know, two images might look the same. I was wondering if you had looked into that in the context of these networks, and maybe what you would expect to come out of those.
We haven't, actually. It's a good point. I would be surprised if they were all of a sudden adversarially robust. It would be, in fact, a big surprise to me, because I think it's a lot of the statistics in the data set and the sort of, you know, having fixed pixel input that, you know, opens the door to these sort of attacks. So I would be surprised if these were more robust, but we haven't looked at that. Yeah, not at all. Thanks.
Thanks.
Wonderful talk. You are a great ambassador for this idea to the broader community, so thank you for that work. I'm definitely really grateful to be able to use your recent review to send to new graduate students, for example, coming into the field. So really nice. In terms of the smoothness in the models and wanting to get around like an artificial, hackish way of building that in, I'm wondering whether the smoothness that we observe in the visual system might be a function not simply of internal parameters, but the distribution of the data that it's trained on. And so you might imagine, for example, training on static images, you're always going to have to hack smoothness into the systems. But there's something about, for example, the natural motion, which is inherently smooth. And so you might imagine training on video distributions could provide some inherent smoothness. One, and two, I think it also really aligns nicely with, as you know, observations that as we move higher in the representational hierarchy in the visual system, it's not just the spatial receptive fields go larger, but the temporal receptive fields go larger as well. So it's like a spatial temporal hierarchy throughout the ventral visual stream, for example.
Yeah.
I'm just curious about your thoughts about trying to bridge the gap between current models and the visual system, not just with the internal parameters, but also training on different types of data.
So it's a really good point. As the lab members can probably all tell you, I'm obsessed with moving this into videos also because my PhD was with Peter König, who with Konrad Körding and others suggested temporal stability as one of the objectives, right? To have a stable code in a changing world so that downstream neurons wouldn't be surprised at every millisecond, right? So that sort of thing on TNNs is very high up on our agenda, if not the highest point on our agenda. We haven't done it yet though. Because the TNNs, mind you, the TNNs are a bit beastly to train in terms of GPU size that they take, because you have local receptive fields and not one convolutional field. So we've got a new rig that we can train on now, but yeah, we had to be a bit wary of that before. Yeah,
it's a heroic effort. I'm looking forward to how it develops.
Wonderful talk, Tim. Thank you so much. It was great. I was really struck by that one plot you had where you had the amount of variance explained in the NSD data set by your RCNN, then MPNet, and then like RCNN trained to predict the kind of MPNet embeddings, and those were better. And you made this point, which is really interesting, that maybe some of the kind of visual information that's kind of percolating in from the RCNNs itself is what's kind of accounting for that kind of boost over MPNet, despite the fact that you're actually your objective is to produce those embeddings. I wonder, is there at some point, when you completely keep trying to predict that, you lose some of that visual information. So imagine if you train your entire model for n epochs. If you kind of test it at like n by 2, you might expect better performance somehow. Have you looked at that?
Yeah. So and I don't think we've looked at that. My prediction going into this, and mind you, we created EcoSet back in the day, which is now when I was in Cambridge still. So that's probably 6-7 years ago. We created EcoSet because I was obsessed with getting away from categories, and I wanted to use fast text and these, then fancy techniques to get at, embeddings. And our point was always to say that there will be an intermediate sort of level where you still have sufficient visual information, but you haven't fully abstracted away into the linguistic space yet. That would be the best. Yeah. And that would be my prediction. But we haven't tested that yet. And another element that is important to realize is that the models predict the captions, but the captions don't cover all of the objects in the scene, right? There's a lot of object information in the scene that people might not think is important to name. So cats spreading out on a laptop would, for instance, ignore the flowers, right? It might be that those things are in the visual system, that RNN can sort of pick that up, but it wouldn't be in the actual semantic target. So even if we were like getting better and better and better, it could still be that the later layers would sort of get at that information, that the language description, which is admittedly short because people do a lot of trials and they give like short, succinct captions, wouldn't cover. Yeah.
Thank you so much.
In the center.
Hi, thank you for this wonderful talk. I think you you made a very good case on how these models can be used to explain perception very well, very impressive, very convincing. Do you think they can scale up to higher order cognitive functions like planning, cognitive control, because the visual system is very much a hierarchical connectionist system, but yeah, more complicated behavior. Do you think that would require the same kind of network architecture, or do we need to move to some other kind of architecture?
I would probably have to tag team Jay onto the stage now, and he would say definitely, definitely yes. No, so what we're trying to do in the lab now is looking more, as one of the projects, looking more into prefrontal sort of computations and trying to understand, you know, putting these things on top of the sort of visual backbones that we train to get at more complex, you know, various tasks that you may want to do and more flexibility, more symbol-like sort of representations. So it's on the horizon, but I don't know. I hope so. Yeah.
Okay, to my right. Thank you so much for an exciting new research sort of area almost here, or at least an outline of a research program. I wonder a little bit how open you are to introducing salient principles of neocortical organization, so more biophysically detailed models, into the model in order to solve some of the issues. So for example, when it comes to the smoothness criterion that I guess lots of people would like to emerge to replace that top-down forcing the network to. I mean, biology has plenty of answers, right? For example, cells that are recurrently connected in layer 2-3 often share the same stellate cell input from layer 4, so the thalamic input is the same, so you get the same selectivity. Or long-range connections, which are expensive, right? But in neocortex, like biology helps itself by having long-range patchy connections. So patchy connectivity is a hallmark of cortical feedback projections, right? Layer 5 B cells reaching back. Really expensive to build that axon long range, but then when you're there, the connection to the neighboring functional column is for free. Hence the patches, which leads also to a smoothness. So I'm just wondering how biophysically interested are you, because we can go a long way into making like spiking detailed networks, right, to solve some of these issues.
Okay. So some of the things you've mentioned are actively explored in the lab right now to see how we can somehow get around this hack, which I don't sleep well because we still hack it. And so that's one of the things we're actively exploring. I find it important that the models remain differentiable, and so that we get that higher level visual phenomena. And that's sort of where some of the things, if you want to do like full-on biophysical stuff, it starts to get tricky. And so that's not where I would draw the line on a principled, for principled reasons, but it's for computational reasons. I think we'll do that. We basically take this top-down approach. We see, okay, what is like large-scale stuff that we can bake into these models to sort of get around this, and we'll slowly make our way down and see what works and what does it, right? So that we don't jump right at the. That's what I had in the. I'm not saying you're suggesting that, but that's what I had in the start. Replicating biological detail for the sake of it will not necessarily give you a big insight into what the individual elements were. So we're trying to do this top-down approach where we bake in more and more stuff. But since we were stuck with like feed-forward convolutional networks, adding recurrence, adding like signs of topography and so on is like the first steps in that direction. But I'd be more than happy to bake more fundamental stuff in. But I'm wary of just copying biology, because you might be copying like the appendix of the brain into your model that the brain isn't using for anything, but you just put it in because the brain did it. And so
you have very sensible constraints, right? You want to keep the ANN alive, like you want to preserve the machine learning benefits that you've obtained, right?
Yeah, so. And I was not suggesting, sorry, I wasn't suggesting you said I should build an appendix into the brain, just to be clear. Thanks.
Thank you. Go ahead.
Thanks, Tim, for a great talk. I find the language modeling work really fascinating and kind of puzzling. But one question I had, though, is I really like the RNN with the different training objectives comparison. That's like a nice tight comparison. The one thought I had, though, is that the multi-hop vector is is binary and very sparse, whereas the sentence embedding is likely a much richer training objective. Mm-hmm. I wonder if another comparison could, well, one, I wonder if that's a concern, and then two, if so, I wonder if like an embedding of just the category label versus the whole sentence might be a different way to get at the category versus sentence level question.
Yeah. Okay, good point. So we've tried to get at the sparseness versus distributedness of the categories by taking the category nouns and putting them into fast text, which is now a distributed language-ish code, and that's not sparse at all anymore. It's now distributed, so that sort of gets at that. So that's one thing. The other thing is we wondered whether we did the whether we do it justice, because a lot of the models that you're training and comparing in the space, as you know, are trained on individual categories that you're training on. So we trained the same models also on EcoSet, which is predicting from image like the single most prominent category, and then looking at the representational layers of those, which are also distributed again, right? And those also don't do as well as the semantics ones. So I think those are two quite good controls to sort of get at that. Yeah.
Just one last question.
Actually, my question is a little redundant because it was just covered, but I wanted to bring it up anyway because AlexNet actually had this regularizer as well. I think local response normalization, which is biologically inspired through lateral inhibition, which is, again, biologically inspired regularizer, which could explain the smoothness constraint. Do you think that could be something possible?
We've played with a lot of stuff. And as far as I'm aware, but I might be wrong, and Adrian can tell you more, but we have tried divisive normalization at some point as a normalizer, and that didn't do the trick, as like a local sort of normalization principle. But again, your guess is as good as mine. We've just played with it a little bit so far.
Thank you.
Thanks.
Okay, I think we'll have to wrap it up now. It's exactly on time, so please thank Tim. And
thank you very much.